contributing to the vulnerability to relapse. We talked about things that are shared by the 3, sometimes 4, if alcohol is included by somebody, addictive drugs. And we talked about things that have occurred, that have been studied quite a bit, like the amp-to-energy ratios and the spines, that really are going opposite directions depending on the addictive drug that the animal's been trained up on. So this period, this enduring addicted state, has some interesting potential, but it's also got a lot of things that are different between addictive drugs. So what about this? What actually happens neurochemically in the nucleus accumbens and electrophysiologically and morphologically when an animal relapses? Most of the stuff I'll show you is cue-induced relapse. So I don't know if I show it or I just talk about it. The heroin stuff was actually an injection of heroin. We started to get away from using cocaine or nicotine or the drug itself in general to relapse animals because we felt there were a lot of epiphenomena that are created by the pharmacological agents. So for example, you give cocaine, in addition to activating this circuitry, this glutamatergic coming in from the cortical and allocortical areas, you're releasing a bucket load of dopamine in the accumbens, which is going to have its own interactions that aren't happening with the cue and that were probably not necessary and may lead us in the wrong direction if we're trying to figure out what's actually mediating the relapse event. All right. So we're going to talk about the relapse event. You guys are familiar with this. I don't think I really need to talk about it. The 2 endpoints we use are the increase in spine head diameter and more AMPA receptors in the LTP versus LTD state. Okay, so this is a study that Meg had just started when she got her PhD and left. Couldn't convince her to stay to finish it. Khaled picked it up as a MD PhD student, and Howay's also played a big role in the follow-up, which was in this paper here. And so the idea is what's happening to this prefrontal to accumbens pathway, and instead of doing a relapse at this point, we just wanted to stimulate the pathway. So I've already shown you some data that talked a lot about, and there's more data than I've shown you, that activation of the prefrontal cortex is necessary for this glutamate overflow that's associated with relapse. And so what we thought we would do electrophysiologically first was just to put a stimulating electrode in the prelimbic cortex and a recording electrode in the core of the nucleus accumbens and stimulate fields. So this is in vivo. The animal's anesthetized, but it's in vivo. And you can see, depending on how much we stimulate here, we can get bigger or smaller fields. And we can do stimulation parameters that give us a very nice LTP. So this is the basal or LTD. This is the basal, and you can shrink it. So we can go both ways very nicely in this in vivo model. It's a little harder in a slice to get the LTP event, but you can get LTP and LTD pretty easily. Okay, so what's happening in this pathway in animals that have been trained up on cocaine or heroin? We've never done nicotine. And this is the data from the cocaine study, but the heroin data look exactly like this. So if you take a control animal and you do the high-frequency stimulation, you get a very nice LTP, and you don't see it when they've been given coke. If you take the LTD and you get this very nice LTD in the control animal, it's significantly blunted in the cocaine-treated animals. And again, this is 3 weeks of withdrawal, so they haven't seen, there's no cocaine on board or anything like this. So initially, actually Anto's lab made the first discovery in a slice of this kind of thing in cocaine animals, and they hypothesized that the cells must be in an LTD. And so it was masking the ability to induce further LTD because they're already in LTD. And that didn't jive with stuff that Marina Wolf had been showing, that there's an increase in AMPA receptors, or Mark Thomas, an increase in AMPA and NMDA ratios. So you have some evidence that it's LTP-like synapses, and then this electrophysiological evidence that maybe it's an LTD because it's masking it. So these data were, I think, very important in showing that it's not whether you're resting here or whether you're resting here that's important. It's that normally you can go between those places depending on how you stimulate the system, but in the addicted animal, you can't. So you have a pronounced limitation on how much classic synaptic plasticity you can produce in these addicted animals. And again, we've done it with heroin and cocaine. And the importance of doing those two drugs is, as you'll recall, the heroin animals, if anything, sit in kind of an LTD-like state. They have smaller spines and a decreased amp-to-NMDA ratio. The cocaine animals sit, if anything, in an LTP-like state with larger spines and an increased amp-to-NMDA ratio. So even though the heroin and the coke animals start in slightly different places on that LTP-LTD spectrum, when you try to induce further LTP or LTD, it's dramatically limited. So one of the megalomaniac thoughts is that maybe this is a major pathology. So you try to put in a stimulus to get plasticity, and normally the prefrontal cortex can teach the accumbens all sorts of things. You can fire it all the way up to LTP or all the way down to LTD. In the addicted animal, you can't push it that far. In as much as synaptic plasticity like this is commensurate with learning, doesn't look like the prefrontal cortex can teach that well. The nucleus accumbens potentiate or depotentiate. All right, so let's go to a more naturalistic situation. Instead of buzzing the animal electrophysiologically, yeah.
So I'm just curious, have you looked at other regions of the TFC, for example, like the OFC, since it does talk to the accumbens as well?
I have not. No, we have not. What we found is that this projection, we did a little bit of amygdala, and a little bit of infralimbic, and the fields, the only place we got really good fields that we were comfortable with, look almost like hippocampal fields that you would generate in those experiments, was this particular projection. So we kind of stuck with that and built it all up. I think perhaps we were hunted a little harder. I know Tony Grace has done a study with fields from the amygdala, and so, but anyway, the answer is no. Okay, so this is stimulating the prefrontal cortex, if you assume the cue is doing that, and I'll show you that it is in a second. Stimulating the prefrontal cortex with a cue to naturally promote glutamate release in the nucleus accumbens, which I showed with the dialysis experiments. You're able to do that. And then the animal relapses. Okay, so this is looking at the amp-to-NMDA ratio in a medium spiny neuron. That's what it looks like in a yolk saline animal. As I mentioned, it's potentiated. This has been seen in a lot of laboratories now. At T equals 0. So this is no reinstatement, basically. 3 weeks of withdrawal from cocaine self-administration. Then 15 minutes after reinstatement, you can see that the system's potentiated. So the amp-to-NMDA ratio is way up. Stays up for 45 minutes, and then it's coming back down to normal, or back to the cocaine baseline anyway, by 120 minutes after initiating cue-induced reinstatement. Okay, if you look at spine head diameter, it looks something similar. So again, there's the potentiated spine. That would be yolk saline and then baseline cocaine, which we call T0. And at 15 minutes of reinstatement, the spine head is bigger, and then it comes back. In this case, it comes back by 45 minutes. So it starts to come back down. So that's an interesting event in itself, is that the AMPA insertion seemed to last a little longer. The spine head is collapsing before the AMPA insertion actually, the AMPA receptors are removed.
Are there changes in NMDA receptor insertion? Because if that
We have not looked at that. We just looked at the AMPA to NMDA ratio. So we haven't done a lot with looking at, for example, surface expression of those two.
Because the ratio could also be used.
Absolutely. Absolutely. And we've done that more so with the nicotine, which unfortunately this is the flagship data. The nicotine has been published, but we did look at in the nicotine situation, both the GLU-R1 went up and NR2A and NR2B, all of them went up during this relapse event. In the heroin, NR2B, but not the AMPA receptors went up. No, sorry, that was baseline. Never mind. Scratch the heroin. So, what was striking about this is the spine head diameter. So the increase in spine head diameter was nicely correlated with the amount of lever presses. Remember, the animals are killed at 15 minutes, so they only have 15 minutes to press. It's not the usual 2-hour session, and so, you know, some really go crazy, others not so much, and some don't even get started. Input-NMDA ratio, same thing, not as nice a correlation, but nonetheless a significant correlation between the behavioral output and the input-NMDA ratio, or the spine head diameter, meaning that this LTP-like event appeared to be correlated with the behavioral output. Interestingly, it's in the supplement of this paper, if you look at 5 minutes of behavior, so the animals are killed at 15, so the input-NMDA and the spine head diameter at 15, but if you just try to correlate it with the first 5 minutes of behavior, it's a very significant correlation as well, implying that potentially this stuff is happening really fast and driving the behavior forward. Okay, so again, just to indicate that the prelimbic cortex is involved, we're in the process of doing this with optogenetics right now, with the ARCH-T type experiment that I talked about. But in this case, we just wanted to find out quickly if the prelimbic cortex was likely involved, so we did the baclofen-muscimol microinjection. And then we came back and did the input-NMDA ratio and the spine head diameter. So there's the extinction rates on the active versus inactive, so this is the day before. Then if we put ACSF into the prefrontal cortex and then show the animal cues, you know, they press on average about 20 times. If we inhibit the prefrontal cortex with a baclofen-muscimol microinjection, you see we get basically extinction levels of pressing. And what you can see is that spine head diameter did not go up. So this is control levels. Did not go up relative to when they got the ACSF, if it was inhibited, if the prefrontal cortex was inhibited. And the input-NMDA current did not go up as well. So by pulling the prefrontal cortex offline, you inhibit the reinstatement and you prevent these LTP-like events from happening. Okay, so data that don't have time to show really in great detail is how did this basic experiment with heroin and got the same kind of thing. He didn't use input-NMDA ratios, he used the fields that I showed you, but he showed that as soon as he gave a heroin injection, you know, the size of the fields went up. So he got an LTP-like event, and he did use spine head diameter. So he showed that, and he killed it 30 minutes afterwards to show that. So it looks like the same thing with heroin. Cassie had this paper just come out showing the exact same thing that I just showed you when cocaine is happening with nicotine when the animals relapse. And Cassie guided this project as well with like an undergraduate student, and they did context-induced, so just to see if, you know, this is a heroin injection, this was Q, just like the cocaine was, and so what if you just have a context? So these are animals that are not extinguished, they're put into the addicted context, and they also show reinstatement and this expansion in spines and increased input-NMDA. Actually, she just did the spines in that. It's a short report, so it showed the increase in spine head diameter. And importantly, we train animals to sucrose, it's a version of sucrose. They don't show the LTP. So you basically show them the exact same cue. They're trained up to take food pellets or sucrose pellets instead of cocaine. Get the exact same cue, they reinstate, and they don't show any of this LTP-like stuff. So it's not just because an animal learned to do something and then they jump up and they're activating their prefrontal cortex because they're seeing a cue that's important and they start pressing the lever. Just like you didn't get the overflow of glutamate in that was a control that we did for that. I guess I forgot to mention it. You don't get the overflow of glutamate. You certainly don't have a downregulation of GLT1, and you don't get this LTP-like event, even though you're getting the exact same behavioral, at least in terms of lever pressing, the same behavioral output. Okay, so what is this? How do we put this together? So there's a loss of an ability to induce LTP or LTD. I mean, that seems kind of contradictory. When they get a drug-relevant cue, they get a massive LTP-like event. When we try to induce it, LTP or LTD, with classic stimulation patterns, we can't get it. So that might be telling us something. So maybe this is what it's telling us. That prefrontal input to the accumbens that's coded for drug-associated stimuli do something different than prefrontal cortex input that's coded for non-drug related stimuli. So the drug-related stimuli are producing synaptic potentiation. The non-drug related stimuli, like the cue, same cue that's leading to sucrose reinstatement, or giving LTP, which is, the animal's not going to, they're anesthetized anyway, but they're not going to pick that up as a drug-relevant cue. We're just trying to stimulate the system. So basically the system is refractory to synaptic plasticity, except when they get this cue that's associated with the drug, and then boom, they blow into this big synaptic potentiation. So maybe this differential synaptic potentiation may be why drug seeking is prepotent over other behaviors in addiction. I mean, it may be very critical as to why the animal has so much trouble controlling this behavior, or the human, has so much trouble controlling the drug-seeking behavior, and actually has more difficulty experiencing things like biological reward and learning behaviors that would allow it to get biological reward. All right, so let's put the tetra into the tripartite. So all we've been talking about is the glia with the GLT1 and the pre and post synapse. So what's this MMP stuff that I've been talking about? And again, what we're looking at here is how the extracellular matrix is involved. And there's just there's a pretty broad literature showing that in order to get synaptic plasticity, the extracellular matrix, especially enduring synaptic plasticity, has to remodel itself. So you have an activation, specifically what most of the work has been done in the hippocampus, of MMP9. So that's the one that's really been studied a lot. And you can look in the learning and memory literature, the LTP literature, and you'll see probably a half dozen papers out there, maybe a few more. Okay, so Alex's, the graduate student's brainstorm was that maybe these are involved in learning and memory as it relates to addiction. And he actually stood on Armina's shoulders a little bit. So Armina was a graduate student whose ultimate paper was this one, that was the heart of her thesis. And that was basically showing that the beta-3 subunit of integrins is upregulated by cocaine. And RGD, which is it's a tripeptide obviously, but it's a ligand for where the extracellular matrix binds to integrins. So nobody knows if it's an agonist or an antagonist. You just call it a modulator because it basically will bind to that spot. So in one sense, it might act as an antagonist because the endogenous protein, extracellular matrix protein, can't bind if the RGD is there. On the other hand, maybe it is stimulating some signaling. And the literature is very confusing on that particular issue. So we don't know, and it's probably state dependent, however you set up your prep, whether it's going to be something that actually signals or it's actually antagonizing. But the bottom line is she was able to give this, and it would inhibit cocaine reinstatement, and this beta-3 subunit of the integrins was upregulated. So that was the heart of her thesis. And importantly, the RGD domain is revealed by metalloproteases. So metalloproteases, when they start cleaving, will reveal this RGD domain that then can signal through integrins, as well as other binding motifs that can signal through membrane proteins. So this was the integrin link, and as I mentioned, there are two, what they call, that are gelatinases, MMP2 and MMP9. MMP2 is constitutively expressed. MMP9 is inducible. Okay, so this is just a cartoon with the idea that you have MMPs, they exist in a proform that's inactive. They can also exist in an active form that's bound by these TIMP proteins. And we're just now investigating a role potentially by the TIMP proteins. They're just basically inhibitory proteins that bind to the matrix metalloproteases. There's something like 25 matrix metalloproteases if you consider the membrane-bound ones as well as those that are in the extracellular space. And there's different TIMPs with different binding affinities. It's a very complicated picture. And I'm not gonna make it that complicated because I can't. I don't understand it well enough yet. We focus on these two MMPs. So the idea is that they carve up the extracellular matrix, reveal binding domains that can signal through these various proteins, integrins being among them. And this is basically Alex's, he looks like Conan, I know, to me he does anyway, but not in real life, he doesn't look quite that much like him. A lot of the work I'm going to show you, virtually all of it, is his work that he's doing as part of his PhD thesis. This is where Armina stepped in and really found that this thing was involved. And Sade has a project looking at thrombospondin, where we found that the receptor for thrombospondin, which is it's an auxiliary subunit on calcium channels called the alpha-2 delta 1 subunit, is upregulated after withdrawal from cocaine. And so she's investigating the role that thrombospondins might play on that. But let's talk about the MMP2. So we can do Westerns as well, but this is really an activity assay that I think is better, because basically if you do a Western, you can't tell if it's bound to the TIMP and inhibit it or not. You can't tell if it's bound to the probe domain, at least not with the antibodies we have. And so you really don't know the activity state. And in fact, MMP at Western measurements, they do not seem to change the MMP2 levels. But the activity is dramatically different. So let's just start here. This is a yolk saline animal. Remember, T equals 0 means it's 3 weeks of withdrawal from yolk saline. And we inject this Fitzy-coupled gelatin, basically. So we inject a substrate for MMPs. And when it digests, when an MMP digests it, it starts to fluoresce. The Fitzy is quenched, and then as it's digested, it unquenches, and you can see the fluorescence. So there's the anterior commissure. We try to highlight that in all of the micrographs. This is a microinjection made. We kill the animal 15 minutes later. And again, there's no reinstatement involved here. This is just basal state stuff. And not much. You know, if I turn the lights way down, you'll see a few of these little speckles in there, you know, scattered around. But there's not very much fluorescence. Withdrawn from cocaine, really big difference. There's a lot of fluorescence. So this MMP2, well, MMP activity. So this is a substrate for MMP2 and MMP9. So at this point, until we get to this corner, we don't know exactly which MMP it is. What's cool is if you look at this a little higher mag, you can see that it's punctate. You know, this is a collapsed confocal image. It's punctate. If you do a new end double labeling, they're all new end. If you do a GFAP, it's not so much around GFAP, it's around neurons. And it looks like when you get this kind of situation, it's the vast majority of neurons. And so it's probably not D1 versus D2. We're setting those experiments up now to determine that. But it looks like a lot of them. And if you look closely, you can see little spots, presumably that are hanging out on dendrites and things, but you can also see the cell bodies tend to get covered, and where we happen to slice a cell body in half, you can see it. It looks like it's pretty much around the outside, like you would expect. Okay, MMP2 or MMP9. So this is the MMP2 inhibitor vehicle. This is if we inject an MMP2 inhibitor, you can see pretty clearly that this fluorescence, if the Fitzy-coupled gelatin is injected with the MMP2 inhibitor, you don't see it. If you put the vehicle in, you do see it. So this is constitutive upregulation of MMP2 activity. And this is this slide shows you basically that there's the saline. Here's the extinction. So we had to recalibrate the confocal because this one was so bright. You saw how bright the other one was. So this doesn't look as bright because we put a lot less energy in to excite the floor. So you basically, the saline looks almost black. What was there, this one now looks something like this. You can still see the track, and you can see the little puff of fluorescence around it. And then here's what it looks like when they reinstate it. So it goes up even more. Here's the quantification of that. Question is it MMP2 or MMP9? And the data are not done. We're in the middle of it. But it's sure looking like it's going to be MMP9. And that fits. The MMP9 is what's inducible. And the MMP2 inhibitor may or may not be doing anything. It's hard to tell. Okay, so could this have anything to do with this LTP-like event. All right, so we're getting all this fluorescence, there's basal activities up, and we get this further increase. It's probably MMP9 mediated during reinstatement. So you've seen these kinds of data. This is the, given a vehicle injection along with, well, just a vehicle injection, so we're going to have the MMP inhibitors here. So vehicle injection, there's the yolk saline, there's the animal that's been trained to self-administer and withdrawn, there's the animal that's been shown a cue for 15 minutes. Okay? So that's what we saw before. AMP-to-NMDA ratio goes up in the coke animal, goes up further at 15 minutes after reinstatement. Okay, here's the inhibition by MMP2 inhibitors of this, and the MMP9 inhibitor brought this down. It's very difficult to tell, you know, whether it's bringing it back to this baseline or, anyway, it looks like both of them work to some extent. The MMP2 definitely shut off this, and it looks like it significantly inhibited that, brought it back to the coke baseline. So MMP2s are apparently necessary for the manifestation of the basal increase in AMP-to-NMDA and the potentiation that you see during reinstatement. MMP9 inhibitor does not appear to be touching the basal increase in the AMP-to-NMDA ratio, but does prevent the reinstated increase in AMP-to-NMDA ratio. So it's preventing the acute LTP-like event that's associated with reinstatement, but it's not preventing the increase in AMP-to-NMDA that you see basally in the cocaine versus the saline animal. Okay, what about the spine head diameter? So again, there's the 3 treatments, and it gets bigger in cocaine and then even bigger during the 15 minutes of reinstatement. The MMP2 kind of wipes it all out, really slams the basal state. And I don't have the ends here, but the ends are all up over 20 neurons. And it looks very similar to the AMP-to-NMDA, actually, and it seems to inhibit the reinstated as well. And the MMP9 probably needs some more data, but it may not be, this didn't come up statistically significant because of the variance in this group here. But it looks like it's probably not, just like in the AMP-to-NMDA ratio, probably not hitting the basal state, but is dramatically inhibiting the reinstated LTP event, or the increase in spine head diameter. You know, I let the slide out, that's too bad. The all-important slide, that, I can dig it out, that either an MMP2 inhibitor or an MMP9 inhibitor will shut off reinstatement. So if you microinject either of those into the nucleus accumbens, the animals do not reinstate to cues. Yeah.
Question, since it's a matrix protease, and does it change the morphology around the synapse?
Is it, so, I'm sorry?
Because in matrix protease, so it digests the matrix. So I'm wondering, is the synapse structure changed?
Yeah, we don't, we haven't done like looking at the size of the PSD or anything. What we look at is I didn't describe the technique. It's you basically make a slice and you inject dye, use a gene gun to inject dye into cells. You've probably seen maybe earlier. It's this lipophilic dye that goes out across all of the dendritic tree. And basically gives you a nice fluorescent outline. And so what we can see is that the spine head diameter is smaller. There's no change in density that we've measured. There's a smaller spine head diameter. We don't know for sure if that's commensurate with a smaller synapse. We're guessing it is, but we don't know for sure. We don't have the markers in there that are measuring that.
Did you look at the transcripts and try to sort of tease out which cell is secreting this enzyme function?
The MMPs?
Yeah, maybe look at
They're
the
glial.
MRA for it's the glia cell complement. Okay.
Like the thrombospondin.
And is it restricted to the nucleocomment region? What about the other?
Oh, the increase, we've done a control experiment in the dorsal striatum,
Yeah.
and it's not happening up there. Okay, so it would appear to be. We haven't done, part of this thesis goes on is to optogenetically turn it on and off and stuff like that. And so we haven't, that'll be the real test of what's going on.
So you've been looking at a lot of postsynaptic readout of glutamatergic signaling, but have you thought about trying to tease apart any changes that might be occurring in the presynaptic terminal, say like release probability, facilitation, depression?
We haven't, we do get spontaneous, in these experiments, we have the spontaneous activity, but we don't have like a TTX mini sort of analysis done. But that would be, it'd be very interesting to look in detail at the release probability to see if it's affecting the mGlu2 signal.
Especially now that you have the optogenetic tools, you can label PRL. Anto talked about this, basically, and how you might go apart looking at some of the presynaptic properties. So, I think that might be a nice way.
Yeah, no, I think it's a great suggestion. No, it'd be a really good thing to do. Yeah, it's, postsynaptic is relatively easy. You just pop it and you get this nice EPSC that you can quantify. And that's, but you're absolutely right, the release probability question. And we do know that the frequency of spontaneous activity, and minis for that matter, is up in the withdrawal period from cocaine. I can't remember we didn't do minis with the heroin, so I don't know, but we did the spontaneous activity. I don't remember what happened with that. So, but it's a really good suggestion. Okay, so this is the summary of the shared long-lasting changes in withdrawal. So you've got that loss of GLT1, you've got the increase in NR2B, and you've got this sort of short-circuiting of mGlu2 photoreceptor regulation, or presynaptic receptor regulation. Okay, so what happens in a relapse event under these situations? And this is sort of cartooning up the data that I showed you. The uptake is diminished, and so you're not going to be taking up as much right at the edge of the synapse, which will cause things to flow out, and you'll start stimulating more of these receptors. In theory, this one is a bit short-circuited, so it's not going to put such a damper on presynaptic release probability, but that's worth testing. We haven't done it in the reinstated situation, and it's going to stimulate these a little bit more. Okay, both of these this is a calcium ionophore, and this will signal through IP to release calcium from the plasma reticulum. The bet is that that's the calcium that's promoting the insertion of the AMPA receptors. As far as the MMP9 upregulation that we see during reinstatement, our preliminary data would indicate that an nNOS inhibitor does a really nice job of inhibiting reinstatement and inhibiting the expansion of the spine heads. And what nNOS does is it's a major activator of MMP9. So basically you nitrosylate the prodomain, which allows it to be cleaved off easier and activate the protein. So you don't really, it's a post-translational event. It doesn't rely on increased synthesis. That's basically, as far as anybody knows, that's a primary mechanism by which it is an inducible enzyme. And it's just a N of 4 kind of thing. The nNOS inhibitor, NPLA, worked pretty well. So we have to go back and look at the nitrosylation state of MMP9 and all sorts of stuff to prove that that's happening. But that's what we're thinking. And the cartoon tour de force, the spine head expands and the animal relapses. Okay, so this will get into some stuff that Robin brought to the lab that I've been kicking and screaming about, but it's actually turning out to be extremely interesting because I wasn't sure we wanted to get into feeding and food reinforcement and really go into understanding that and try to get to how does the pathologies that we see in overeating potentially relate to drug addiction. And at least the animal model that we set out with gave us some very interesting insights, I think, as to what is an addiction-related adaptation and what is an adaptation related to sort of normal variance biologically. And it fits, it lays on pretty well with some of the stuff we've been talking about. So here's the spectrum again. You go from social use up to compulsive. And so swinging between these two probably has a lot to do with just biological vulnerabilities. And so some people are going to become a little more compulsive about their drug use than others by virtue of inherent or epigenetic vulnerabilities that they have. Other people, it may be a vulnerability thing, it may be that any of us that beat on our brains hard enough with an addictive drug will ultimately push ourselves here. But the idea is that this switching could be well within normalcy. This switching is pathological. And so maybe using a feeding model, we might find things that we think of as drug-induced adaptations that we might be able to assign to this zone because it's happening in kind of the normal population if we just isolate out the overeaters. Whereas the things that are drug-induced will only be happening here and not in that group. So it's a perspective that we just started toying with like 10 days ago, 2 weeks ago. I mean, when we started getting Robin's data really put together and we started thinking about the potential significance of what she's finding. So this is basically, I think she explained this, so I'll go through it pretty quickly. She has them eat a high-fat diet. Some guys get overweight, some guys don't, and that differential, you can see it in kilocalorie consumption as well. Take the top third and the bottom third and start studying. So the most resistant and the most prone. And if she does a little electrophysiology, oh no, sorry, this is the behavior. She does the behavior on it. You don't see much in the early FRs, and this is just pressing for food pellets, sucrose pellets. When you get up to an FR5, you can see that the prone guys look like they're willing to work a little harder. So FR5 means they get a reinforcement amount every 5 lever presses. And these guys get a little more excited and motivated about pressing for it as you start making it more difficult to get the drug. And if you do a classic progressive ratio, where you just increase the difficulty of getting the pellet, they have to press more and more times until eventually they stop. That's called the break point. And you can see that these prone guys have a higher break point than the resistant guys, implying that they are willing to work harder, that they're more motivated to get the pellet. Okay, so it fits with this population. So now we've got a population of highly motivated animals to go get food. So what are some of their characteristics? And these are just, we're just kind of getting into the beginnings of this stuff. But they don't show LTD. So the prone animals are not doing a very good job of, we aren't inducing, this is in a slice now, this is not that in vivo model. We're not getting good LTD, we get nice LTD in the resistant animals. If you look at the AMP-NMDA ratio, they're up here, the resistant animals are down there. So this characteristic of reduced LTD and increased AMP-NMDA ratio that we see in the coke animals, or the nicotine animals, if we just break our population apart, we can see that. So this may be, the good news is these may be vulnerability characteristics to being highly motivated and sort of overcooking yourself on something like food, but these are happening on their own. You might, those of you, there's a Piazza paper out, came out in Science in 2010, where he trained animals on cocaine for 3 months, and then some of them would actually, after 3 months, a subpopulation would go through stress to keep getting the drug, and the others would just drop off when you start stressing them. And so he looked at that group that would go get the stress, that would go through the stress, and this is what they showed. So possibly, all he had to do was give them high-fat food, and he could have isolated out the guys that would have ultimately gone through the stress too. Point being is that this sort of change in the direction of highly motivated seeking is, we see it with drugs, and we see it in this model of obesity-prone animals. But there is no effect on the glutamate transporter. glutamate transporter is rock steady, which fits with even when we just did regular sucrose training and then did our reinstatement on sucrose, we didn't see any glutamate overflow. We don't see the induction of this AMPA-NMDA ratio that we're hypothesizing is mediated by this glutamate overflow to these extrasynaptic receptors. We don't see that stuff, and they don't have the downregulation. So, is this downregulating by the addictive drugs? Is this what's really pushing the pathology? And the other stuff, AMPA and NMDA up or down, possibly spines kind of can grow and shrink just like they're up for coke and they're down for heroin. And that's telling us something, but that's not sufficient to really create an addictive state. What's required is that the synapse can't contain its glutamate, and it flows out and does a big stimulation of these synaptic glutamate receptors in response to that particular stimulus.
Maybe I missed it, but have you taken these animals and then looked at rate of self-administration?
We have not.
Okay.
No, it'd be a great experiment. I mean, the hope, of course, would be is that because we're looking at a motivational state probably, that they would learn to self-administer more, and then of course they would become addicted because that would theoretically drive down the glutamate transporter. So we could probably take with cocaine. I don't know. I would be interested to do the experiment the other way too. We take them to cocaine and see if they were more prone to the high-fat diet. So there's a lot to do with this. Okay, so finally, let's end with a potential pathway for using all of this stuff to cure addiction. And I'm not walking on the Appalachian Trail or flying down to Buenos Aires to see a mistress. But maybe we can still do something. Okay, so here's, you guys are familiar with this cartoon now. And here's the ways that things have been done in animal models. So the data we have that these adaptations that I'm showing you may or may not be able to influence reinstatement behavior and possibly human relapse. So restoring GLT-1 with N-acetylcysteine, ceftriaxone, or propentophyllane. This is a new one that Kate Reisner in the lab, she's now just took a job at UNC in the Department of Psychology there. And that's a paper that was just sent back in with some revisions. But all of these drugs will restore glutamate transport. And they've been characterized for other reasons. They all inhibit reinstatement, whether it's to heroin, whether it's to nicotine, or whether it's to cocaine. And I'll show you examples of that data. But there are other things. So if you inhibit NR2Bs, this paper's out now in 2013. Heroin or nicotine, if you block those with a relatively selective NR2B antagonist, or in the case of the heroin, we actually downregulated it with an siRNA, you prevent reinstatement. So supporting the idea that the glutamate has to come out and stimulate those receptors to reinstate. The NR2B antagonist in the heroin paper also prevented the expansion in spines and prevented the LTP glycogen. So it prevented the commensurate LTP. That was not done in the this particular paper with nicotine. And it's never been tried with cocaine. It's just kind of backwards from how we usually do things. mGluR5, the literature is chock full of papers showing that mGluR5 antagonists, they're actually allosteric modulators, but they're negative modulators, will inhibit alcohol, nicotine, cocaine, and heroin reinstatement. I just listed a couple papers, but there's a bunch of them out there if you Google it. And you can either downregulate the rise in AGS3 to restore signaling, or you can give an agonist, an mGluR2 agonist, and you will inhibit heroin, nicotine, cocaine, or alcohol reinstatement. Okay, so the animal model is good. I mean, it shows that all of these things that we've been talking about can potentially manipulate reinstatement. Sometimes the manipulations were done directly in the nucleus accumbens, sometimes they were done systemically, but most of them have at least some of the experiments done directly in the nucleus accumbens. Okay, so this is just to show you that Both ceftriaxone and NAC bring back GLT1. As I mentioned, we used to think that it was the cysteine glutamate exchanger because we were basing everything on our cocaine data. And we started using NAC because NAC is known to activate the cysteine glutamate exchanger. And so we set it up and we got great effects with NAC as N-acetylcysteine. I'm not going to show you the experiment, but if you hold GLT1 down with an antisense, when you give NAC, NAC doesn't work anymore. If you hold the cysteine glutamate exchanger, we target the catalytic subunit, XCT. If you hold that down with an antisense, NAC still works fine. So it has to bring back the transporter, not the exchanger, to inhibit reinstatement. And ceftriaxone also actually has an effect on both, but it was characterized as a smart GLT1. Okay, so these are just examples of some studies that have been done showing that these drugs work in the reinstatement model of heroin. There's a nicotine one from Athena Marcou's lab, and there's Lori's work with ceftriaxone in cocaine. Okay, so N-acetylcysteine has a venerable history in both almost folklore use as an antioxidant and in normal clinics. It's used as an a mucolytic agent. You can inhale it, and it's also used for acetaminophen overdoses, so they give you a big whopping amount. Now, the reason for that, which I didn't go into because we don't think the cysteine glutamate exchanger is the relevant action anymore, but N-acetylcysteine is a substrate. It gets taken in, it's formed into cysteine, which then makes glutathione. So if you are overdosing on acetaminophen, your liver basically gets scrubbed of glutathione. And you need glutathione as part of some major sort of redox balancing agent inside of cells. So you start suffering oxidative stress, your glutathione levels get too low and it can become toxic to the cells. And so one of the first lines of defense is when somebody's overdosed or nearly overdosed on acetaminophen, you load them up on N-acetylcysteine to bring the glutathione levels back. So anyway, it's available. It's out there. You have to take, in this study you can see, it's not trivial. You've got to take 2.4 grams a day. And because it's got a disulfide linkage, and sometimes that'll break, it stinks. So it smells like rotten eggs, basically. It smells like sulfur. And so we've learned to package it under nitrogen, which prevents it from breaking. It sort of stabilizes it. And what we do is the placebo, we paint on a little bit of stink onto the placebo pill, so that when they take it and if they got a little bit of that sulfur taste in their mouth, they'll get it from both. Okay, so that's how we handle that. Otherwise, they would know. Anyway, this is Kevin, and he's a clinician over in psychiatry, and he did this very interesting study with adolescent marijuana users. And the bottom line is they took N-acetylcysteine or placebo for 8 weeks, and they had more negative urines for THC in the N-acetylcysteine versus placebo. If you look at this, it doesn't look like that much, but this looks a lot like how well Prozac works in a depression trial or something like that. I mean, it's in the ballpark of how far you can bump people in the types of clinical trials that we did. This is more dramatic, but it comes on top of a giant negative study. So this is Bob Malcolm, and we set up this study where we had 3 arms. So there was a high dose, a medium dose, and placebo of NAC. Cocaine addicts came in, we took urines for a week, and then we put them on N-acetylcysteine. They had to come in and give urines twice a week, and they're out in the wild otherwise. There's no, they get a little cognitive therapy when they come in to get their urine and get their next week's worth of medicine. The first thing we realized is only half of them met criteria for even taking the N-acetylcysteine or placebo. It wasn't like they didn't like the N-acetylcysteine. They didn't take, about half of them didn't take placebo. They had that basically have it. We can measure, we doped the pills with riboflavin so we could measure it in their urine. And they had to have it in there 75% of the time, we felt, in order to, that they would have been fairly continuously dosed. and only half of them made it. So we went from 150 down to 75 right there. And we got a little bit of a signal. So probably not quite as good as what we saw with the marijuana there. But there was this, we got to put a few stars on the curve. But what we realized is when we started, digging into the data, as one does when one really wants to prove something is true, and the actual first analysis didn't quite show what we wanted, we started looking at, we came up with a very interesting observation. So if you take those that were abstinent going into the trial, so their urines were actually negative the week before we started them on the N-acetylcysteine. And then you just look at relapse rates. So you don't look at how much benzoyl-acognium the coke metabolite is in the urine. You just look at whether there's anything in the urine. If there's any coke, they're considered relapse, then that's the end. So you create a survival curve, basically, which is what we've got here. So what you can see, this is the 8 weeks, and virtually everybody relapsed that was on placebo, and they did it pretty quickly. There's the 2 doses of NAC, and you can see we had a lot less relapse. Now the N is, because we kept trimming it down, is very low. But this, even with that low end, it was statistically significant that the NAC groups relapsed less than the placebo. So we've now set up another trial where because the VA, is very, we don't inpatient addicts very much in the United States, as you're probably aware. It's just, unless you've got a lot of money, and then nobody likes to do clinical trials on the clinics where people are spending $20,000 to go into the clinic. So it's very difficult to study abstinent people in the United States. So what we found is that our VA has a 4-week sort of intensive cognitive therapy session where they actually do urines and they get thrown out if they come up positive. So they only stay in that if they've been abstinent for 4 weeks. So we pop them on the NAC at the very beginning. And then the hope is then when they go back in the wild for another 8 weeks, that we'll get something like this, only with maybe even better effect, or certainly with a larger N. So that's kind of where we're at in trying to develop these clinical trials. And these are some of the studies that have been done with NAC. Just point your attention to, it's been tried in cigarettes and coke and marijuana with some, small positive effects akin to what I showed you with the marijuana. This is what makes it interesting and makes one wonder if somehow this N-acetylcysteine bringing back GLT1 isn't getting at a very fundamental substrate of what we would call compulsive anything. Compulsive gambling, trichotillomania is compulsive hair pulling. Maybe that's a way to target a lot of obsessive compulsive types of behaviors. Anyway, there's a couple positive trials with these 2 non-drug related compulsive behaviors. All right, so very quickly, I just, I'm supposed to impart like a little wisdom to you guys. And so there's this old Greek saying, and it's actually in an epic poem, and there was a general Kalivas, is how you'd say it in Greek. And he had this saying that's, you've heard other people using it too, but according to my family lore, this is the first guy who said it, and it's fortune favors the brave. And I shouldn't tell you how the story ends, because that was part of his pep talk, and then they all got wiped out by the Persians, like the next day. So, don't listen to that last part. What you have to pay attention to is that in order to get ahead in the business that we're in, you have to have courage. You're going to get a lot of rejections and things like that, and just keep at it, because if you're doing good science, we still live in a society that actually very, very highly values science. Anybody in our society, almost if you interview them, will say that yes, they think science will ultimately solve this problem or that problem. So people really do believe that science is a road towards advancement, whether it's a better iPhone or whether it's curing cancer or schizophrenia or drug addiction. It is something that's worth exploring, worth funding. And we've seen it, we have seen a drift away from that a little bit, of course, that everybody's been feeling it. But we still have major research universities like no place else on the planet. All the universities are still there. That's why they're just going to have a bunch of empty labs. You know, the funding will still be found to develop new ideas and research, and you guys have every opportunity to be part of that. It's a little more difficult now, and there's a little more, certainly more fear, a bit more of a bunker mentality. But just to sort of bring a little light in, the people that were really good at getting grants and who still have them, they'll be gone in 5 or 10 years. You know, they're all in there. They're like me. You know, they're in their 60s. They're just 60, not just in the 60s. 60. And you know, so for another 10 years or something, I'll be kicking around trying to get funding and competing with you guys. But that won't last forever. And you know, money's going to open up because there is a lot of money being held in that, and we know how to write grants really well. You know, we've been doing it for a long time, so it's tough competition. The NIH has made, I think, really important strides towards funding new investigators. They've changed funding lines, et cetera, and so it is actually harder for us, funding-wise, you know, percentage-wise, to get the grants. And so it's, you know, efforts are being made to preserve this generation of scientists, you guys, and I think they're successful. I believe that, you know, if you are doing good science, you guys will do fine. So don't lose hope. Be strong, and I think it'll work out for you. There's another saying that's kind of along these lines, and it's, you know, I don't know what you would make of this. Don't take it literally. It's what you guys know the story of Zorba the Greek. So basically, it's this uptight English guy comes down to Greece, and he just can't understand that Zorba's trying to explain to him how you're supposed to live your life, which is basically just putting it out there. You know, just do it if you think it's right. Do it if it feels good, and you know, which is obviously the intent of that statement. And the end of the story is the Anglo guy learns to dance, basically. So he dances, and that's kind of the end of the story. You've really got to just take chances with your ideas. I mean, it's okay to be incremental in your data. You should be. You know, you can't. Your data has to sort of build. You know, you're building your edifice, but it's okay to shoot beyond that. Now, you don't have to become megalomaniac or anything, but really to shoot beyond it. Think big about what's possible, and then if you don't get it, you know, if it doesn't go in that direction, that's no big deal. You can even write it down in a discussion. And if it turns out not to be right, hopefully the data's right. You want the data to be right. But if your extrapolations aren't right, that is no big. That is really not a big deal. You know, you want to explore what's possible with your data. You really want to. And I think that's, if you do that and you're kind of a believer in your own data set, I think you're going to find things more frequently. Same thing goes with where you want to publish. I mean, if you're not getting rejected all the time, you are not sending your stuff to good enough journals. And so you really just, you know, push. All right. That's about all I do anymore in the lab. You know, I don't do much of anything. I just sort of sit there and watch everybody. These are the people that bring you everything. And I've introduced some of them. Two of them are here, obviously. And we have collaborators. The one group that I want to point out very closely is Gary Aston Jones, Antonio Lavin, Bob Malcolm, you saw him as a clinician, J.K. McGinty, Art Regal, and Ronci actually just left. This is part of our center grant, and these are the people that, you know, we just bounce ideas off all the time. And it really creates a critical mass for us to be able to sort of test out some of our weirder ideas even before we put them in the discussion papers. You know, and I would, if you're interested in this field or whatever you're interested in, it's a technology, which is your stage of development, just being interested in pushing a technology is what you should, you know, that's a great thing to do. But it's important to have a critical mass if you can find it. I mean, sometimes it's, you know, the only job available there isn't the critical mass, you get to be it. I was at Washington State University for many years as the critical mass, you know, and we did fine. We did fine. And so you can do that, but it really helps to have people that have been thinking about this a lot and bounce ideas off. And if you don't have them there, find them at meetings, like I was talking about with Bob Post. I mean, that's when I was at the vet school in Washington State University. I mean, I found these guys at these meetings and really, became good friends with them. And it's very important. You can't think of it all yourself, and you need people to bounce ideas off of. All right, that's it, my talk. Thank you very much.
So you mentioned these targets that are somehow involved in reinstatement, like the NR2B and blue Rs. Is that related to the changes in the glutamate transporter function, or do you think it's completely separate?
No, I think that's the whole point. I think they're intimately linked. So I think that you don't access the NR2Bs and the mGluR5s in a pathological way unless you're getting that extra glutamate out there. I think that you're getting it's the overstimulation of those receptors that's producing this rapid LTP. We have to prove all that. They saw the little curvy arrows. And so, but that's our game plan right there. Is what you saw.
So are you familiar with the Dan snare mice that have attenuated?
Yeah.
So Julie Glendy just showed that those mice don't reinstate either.
Right.
And
But
we've shown
It's with the GFAP promoter, right?
Right. Yeah. So and we've shown that they also have reduced surface expression of NR2B. So that's why I'm
Interesting.
obsessed with that target.
So that could be more of a. So they're not secreting as much glutamate theoretically, right? That's the idea.
Yeah. Well, we've
I
shown
mean,
it
that's
to
with
be
the DN
linked.
snare you would predict that would have less vesicular.
We've shown it to be linked to reduced ATP or adenosine activation of A1 receptors.
Oh, okay.
Not in the cocaine model, but separately.
Okay, yeah, no, I wasn't aware of that. So just in the resident knockout mice, I think that's really a very interesting finding, and we'll have to think about it as we go through our model. But the resident the constitutive knockouts, they have a compensatory response. It's not just even developmental, but even as adults, that may or may not reflect what an intact animal does in response to drugs. That would be a warning that we went through our phase of constitutive knockouts in collaboration with Paul Worley, who has a knockout for every PSD protein, and we got into the homers quite a bit. And as we started going through it, we realized that every constitutive knockout of a significant postsynaptic density protein had some kind of phenotype for cocaine. A little more locomotor activity, a little less, and so we started thinking that if you take stuff out that's absolutely critical, you're going to change things, whether you're proving that it's critical for the cocaine effect or not. It's hard to say. You basically stress the system. The system's missing this protein. It's not going to have the ability to compensate, and the behavioral output will be different. Now you're not looking at a behavioral phenotype.
These are actually inducible anyway. It's not constitutive.
Oh,
So
okay.
they
I didn't know
develop
that. All right.
But yeah.
What's interesting, we've done something similar where we used dreads. So we have a GFAP coupled GQ, and when we stimulate that, it inhibits reinstatement as well. And so in theory, that's kicking out something. But of course, first thing we think of is glutamate. So what we're doing, and we're about halfway through this experiment before we publish it, whether it works or doesn't work, we're still going to publish it, but we're looking at whether mGluR2/3 receptors blockade will inhibit the effect, because we're betting that it's glutamate coming out and stimulating those presynaptic receptors to shut down glutamate release. So it's a similar experiment. We haven't looked at whether blocking NR2B receptors would prevent that. That would be something of interest. That's a very nice point. Thank you.
Have you tried giving the GLP-1 inducers, either ceftriaxone, NAC, or PPF, during self-administration training, and then see if it would block relapse by preventing GLP-1?
Right. No, we haven't done that. Since the GLP-1, we've realized that GLP-1 is important. So David Baker, however, did that, not when he was working with us, but on his own later. Gave NAC and then showed that it prevented the regulation of the cysteine glutamate exchanger, the cocaine-induced regulation of cysteine glutamate exchanger. And it prevented because he did a long access paradigm, so it did not inhibit regular self-administration, but it prevented the escalation of the self-administration. And so he was linking those two together. But we haven't, I mean, it'd be a good experiment to do, to go back and find out if we can prevent the GLT1 thing from ever happening. He also prevented animals from reinstating, by the way. So given what we've been saying about GLT1, we would predict that it prevented the GLT1 from being downregulated, but we haven't looked at that. It's a very good question. We have given chronic NAC during, I didn't go into it, but a lot of the data that you saw there, NAC was given chronically during extinction, and then some of those data were generated like 2 weeks after, so there was no NAC around. So there's, in terms of restoration of AMP and NMDA ratios and stuff like that, it's a relatively enduring event, so there's no more NAC on board and they're still seeing it. But again, that was all before the GLT1 awareness. We haven't gone back and done all that and seen if GLT1 comes back.
So do we even know if GLT1 downregulation is happening during drug exposure, or
Do
is
not
it
know that.
absence
Don't know the
related
answer,
change?
yeah, that's a very good question.
Yeah, this might be like a little sort of out there idea. So you look at the astrocyte, and you show there's oxidative stress, and it's nice if I have this NAC. I mean, if there's oxidative stress, the microglia could be activated. And as we know, from Barrett's group and Beth Stevens, these activated microglia then can actually go in and alter the synapse through like synaptic stripping. So I wonder, have you looked at the microglia potential role in the kind of a synaptic
No, you know
remodeling?
who's doing that? I mentioned Kate Reisner, actually. Where's Kate? She's in this one. Yeah, the woman with the glasses in the very back, looking out this way, that's Kate. She just took a job at UNC. That is her project, is to take these glial markers, both microglial and extraglial, and really evaluate those as it pertains to addiction. So we're just stick with the GLT1 for now. And she's looking at all sorts of other stuff. So it's a very, it's a very good idea, because I think that she'll probably find some things in there.
Does NAC decrease like drug toxicity overall, like not just the doesn't just the GLT1 effect, but you said it has more systemic effects too, like glutathione?
Yeah, but the interesting thing with the glutathione is if I took you, because it's been used in lots of different trials, like AIDS trials, for example, because you have low glutathione, and now they don't have to worry about it within the drug. And what they found is that the normals, given N-acetylcysteine, glutathione levels don't go up. It's like they're already kind of optimized, and they don't go up. But if for whatever reason your glutathione levels are down, then they come up. What we found is that when we, again, this is back in the cysteine glutamate exchange days, where we looked at cysteine glutamate exchange in other brain areas, and N-acetylcysteine would not affect that because it was normal, but in the nucleus accumbens it would bring it back up. And so, it's almost like magic, right? It sounds like the perfect drug. It only affects the pathological situation, and there's some kind of homeostasis if the system is healthy, and the N-acetylcysteine is just metabolized. It's just like taking vitamin C or something that you'll urinate out if you don't actually use it. And so it's kind of like that with the N-acetylcysteine. So it has the potential to be a really good drug in that regard. Its bioavailability is horrible, as you can see.
So we just had some recent data on GLT1. You got trained on the sugar reinforcement for 3 months, and you got downregulation of GLT1 in the dorsal striatum, and actually N-acetylcysteine prevents animals from seeking the drug at that time.
So this is, I missed the first part of what you said. That's an experiment that you...
With secondary sugar reinforcement for like 3 months.
Okay.
So we had a paper in Addiction Biology last year in which we showed that N-acetylcysteine decreases cocaine seeking when it's well established. And we've just probed that you have downregulation of GLT1 in the dorsolateral striatum in these animals that is normalized by N-acetylcysteine.
This is nicotine?
No cocaine.
Go with cocaine, okay.
And we're just wrapping up a paper that the same thing happens with heroin.
That's fantastic.
So that's
And N-acetylcysteine brings it back.
Yeah.
So you basically, when are you giving the N-acetylcysteine again? Sorry.
it's just after 3 months of self-administration.
So they take the drug for 3 months,
and then you give N-acetylcysteine prior to seeking sessions.
Okay, and it inhibits the
And
drug
it
seeking, so it's an
decreases drug
acute
seeking.
injection of NAC.
Yes.
And then that also prevents,
Yep.
wow, that, just one injection restored
Yeah.
it. Fantastic. So we don't have to give it to them for 10 days to get it all back.
And actually, we had another interesting result, which is when you give rats extended access to cocaine, they start escalating, and they've been escalating for 2 weeks. Then you start chronic treatment with N-acetylcysteine. It doesn't bring back escalation.
Okay, so it
It
does
doesn't
not
break it.
persist.
So it prevents the development of it. We have the same, but it doesn't prevent the
Okay,
expression of it.
Yeah.
But then, if you punish the animals, they extinguish, and you stop punishing them. Those treated by N-acetylcysteine never come back.
Okay, okay.
If they're given the drug is there, they just won't take
They
it.
just don't take, they don't go back up to the escalated level. Wow. So it is magic.
Yep.
That's good. Yeah, that's amazing. That stuff has been published, or
No,
just the first part?
Yeah.
In addiction biology? All right. Thank you.
But you have the clinical trials, right? You're doing clinical trials in this, right?
Yeah. When NAC was so available, we could just
Yeah.
jump to clinical trials when we thought it was a cysteine glutamate exchanger. So why do anything else with it, right? Well, we want to find out how it works, because theoretically, we can target it a little better. NAC does a lot of stuff.
So were there any good results from that?
With the NAC? Well, just what you saw.
Yeah.
It inhibited marijuana smoking,
Yeah.
and in that subpopulation,
But you had some with cocaine, right? You were doing.
we did with cocaine, right, and
Yeah.
that's the big trial. We just got a very small signal, but when we isolated those that were abstinent going in, they didn't relapse nearly at the same rate that the others did. It was I think the very last slide. But it was a small N for going back and reviewing that trial in a big way.
It sounds like there may be some subsets which would be sensitive.
Yeah.
And did you look at GLP-1 expression in other brain regions, like the hippocampus, for example? Animals?
We actually haven't. No. How could I? I don't think we have. Coty did a little bit. She did some uptake in the dorsal striatum, but that was with a stress model.
Yeah, I mean, I would be curious about it within the context of abstinence-related negative affect. And GLP-1 levels, at least in humans, have been shown to be downregulated in depression. So I would be curious to see in
And where were they downregulated? In the
In the
hippocampus?
hippocampus.
Okay. Yeah, it'd be very interesting. Yeah, we've actually Schofield, kind of guy that looks like a logger with the glasses on. He's actually setting up a PCR for other reasons, and he's agreed that he will, and he's going to do a bunch of brain areas, and he's agreed to at least PCR for GLP-1 all the way around. So we'll maybe have at least at the mRNA level, we'll have some answer. But no, we just haven't done that. It needs to be done to isolate it. We've done the cysteine glutamate exchanger, like I said, and that seems to be fairly selective for the core of the nucleus accumbens. All right, well, thanks a lot.